Greetings, everyone. Our project title is Efficient Blood Cell Detection with DETR, a novel approach for automated hematological image analysis. My team members are Jayasurya and Janvi Kulkarni, and myself, Lalita. Our project ID is PC26, and our guide is Dr. Ravi V. Sir. Blood cell detection is an important aspect of medical diagnosis and treatment. Blood cells are an essential component of the circulatory system and they play a critical role in carrying oxygen and nutrients to tissues and organs throughout the body. Abnormalities in blood cell counts or types can indicate the presence of various diseases, infections, or conditions. Therefore, accurate and timely blood cell detection is required for diagnosing and treating a range of medical conditions. There are various methods for detecting blood cells, including manual examination under a microscope, as well as an automated analysis using specialized equipment. In this research, we present a new technique for identifying blood cells using transformer models, specifically known as the DETR, detection transformer model, which has not been extensively studied in the biomedical field. The objectives of our project include to provide an overview of the transformer model and its architecture, to explain the DETR algorithm, a cutting edge object detection model built on a transformer architecture, and to implement the DETR on the blood cell image dataset and provide details on the training process, hyperparameter tuning, and any modifications made to the original DETR model for the specific task of blood cell detection. The other set of objectives include to analyze the architectures of DETR and YOLO V5 and provide a comprehensive analysis of their performance on a benchmark dataset using inference metrics such as mean average precision and IOU scores. To provide a detailed explanation of the technical details and working principles of both the models, evaluate their accuracy, speed, and computational requirements, and analyze the impact of hyperparameters on their performance. The dataset that we have used in this study is the BCCD, Blood Cell Count and Detection dataset, to perform the blood cell detection using both the models. The main framework that we have used is the PyTorch, and the platforms where we have implemented the models are Jupyter Notebook and Google Colab. This is the block diagram of our proposed method. First, we have collected the data from the dataset known as the BCCD, and we have augmented the dataset to increase the number of images in the dataset. Later, the dataset is split into train, testing, and validation, which is further processed into training the YOLO V5 and the DETR models, and the outcomes are observed and compared. Now, let us talk in detail about YOLO V5 and the DETR implementation.
The YOLO V5 stands for You Only Look Once, proposes using an end-to-end neural network that makes predictions of bounding boxes and class probability all at once. YOLO algorithm employs convolutional neural networks to detect objects in real time. As the name suggests, the algorithm requires only a single propagation through a neural network to detect objects. This means that prediction in the entire image is done in a single algorithm run. YOLO V5 uses a new method for generating the anchor boxes, called dynamic anchor boxes. It involves using a clustering algorithm to group the ground truth bounding boxes into clusters and then using the centroids of the clusters as the anchor boxes. This allows the anchor boxes to be more closely aligned with the detected objects of size and shape. So the architecture of YOLO V5, there are two types of object detection model. two stages, that is, object detectors and a single stage object detector. The single stage object detector architecture are composed of three components: the backbone, neck, and a head to make dense predictions. The backbone is pre-trained network used to extract rich feature representation for images. This helps to reduce the spatial resolution of the image and increases its feature resolution. The backbone network consists of a series of convolutional layers followed up by activation function, pooling layers, and skip connections. These skip connections are used to combine features from different levels of network to capture objects at different scales. The model neck is used to extract feature pyramids. This helps the model to generalize well the objects on different sizes and scale. The model head is used to perform final stage operation. This applies anchor boxes on feature maps and render the final output. The final output are classes, objectness scores, and bounding boxes.
This is a novel approach, DETR model. DETR model is a transformer-based, flexible, end-to-end trainable object detection model, allowing the model to learn directly from the object detection task and avoid the need for intermediate representations using a self-attention mechanism. The DETR model takes the whole image into a single consideration, and the model processes the entire image using an encoder network, typically a convolutional neural network, CNN model, to extract a set of feature maps. These feature maps contain information about images at different level of abstraction. Here in our block diagram, our model uses ResNet 50 architecture as CNN backbone for feature extraction to generate 2D feature maps, which are then flattened into 1D sequence of visual tokens. They are also known as initial raw hidden states, which are then processed by a feed-forward network called a set prediction network, SPN. In the encoder, the SPN takes the raw hidden states as input and generates a fixed size set of object queries. It is visible in the encoder with the boxes. Each object query is associated with a learning class token that corresponds to the object class. Next, the SPN and the transformer joins and attends to the raw hidden states to refine the object queries and predict the final set of object detection, including the labels and bounding boxes. Here, the position encoding represents the spatial information about the image and which are added to the object queries to enable the transformer to reason the relative spatial relationships between the objects in the images, resulting in the final input vector or the final set of object detections. The bounding box are generated using these final set of object detection.
Now, let us observe the results that we have obtained after training the YOLO V5 as well as the DETR model for the blood cell images. We have evaluated the performance of our model using standard metrics including precision, recall, and mean average precision, known as the MAP score for each blood cell type.
The performance of a model was using standard matrices, including precision, recall, and mean average precision for each blood cell type. The MAP score was calculated by comparing the detected bounding boxes with the ground truth bounding boxes. The bounding boxes uh, loss was used to assess the accuracy of the model in locating the center of 46 objects, while the objectness loss indicated the probability of an object existing in the region of interest. Our model is run for 50 epochs. The model architecture consisted of 232 layers with 2 lakhs 51,912 parameters. The computational time was 4.5 minutes. Um, as in these graphs, the x-axis shows the number of epochs the model has trained, and the y-axis is the metrics. for the box loss versus number of epochs. As the number of epochs increases, the box loss decreases. The box loss, the object loss is also the same. As the number of epochs increases, the object loss decreases. And it is the same for precision. As the precision increases, as the number of epochs increases, and the same goes for recall and MAP. The graph shows that after 50 iterations of the model, IOU score is 0.88, and this score consistently rises whenever the loss takes a dip. The MAP score is calculated by comparing the detected bounding boxes with ground truth bounding boxes.
Here in DETR results, the predicted blood cell types are visualized by overlaying the boundary boxes on the input images. This is the training image output. For the results, we are plotting the loss and MAP scores. In loss plot, the x-axis represents the loss value and y-axis represents the number of epochs. In the MAP plot, the x-axis represents the MAP value and the y-axis represents the IOU scores. You can observe that with the decrease in loss curve, there is an increase in MAP scores and precisions. The maximum MAP score achieved by DETR model is 0.89, which occurs at the IOU threshold of value 2. The loss plot is separated into three types. The loss CE, which is cross entropy loss, and B box, which is boundary box loss, and the GIOU loss, which is generalization, intersection over unions. The x-axis represents the loss value and the y-axis represents the number of epochs. The model undergoes a substantial improvement in terms of loss reduction and MAP score, enhancing during the first 10 epochs, followed by a more title change in subsequent epochs. The graph shows that the loss function decreases during the training process and monitors the performance of the model by adjusting the training process wherever required. These losses decreases over time as the model becomes more accurate at detecting the objects in the image. The MAP score measures the average precision of the model across all the class of objects in the image. This is the testing image. The more the MAP score indicates that the model is better at detecting objects in an image.
In terms of architecture, YOLO V5 follows a one-stage anchor-based approach where bounding boxes are predicted directly from predefined anchors at multiple scales. DETR adopts a two-stage anchor-free approach where objects are detected directly using a self-attention mechanism in a transformer-based architecture without relying on predefined anchors or region proposals. Another significant distinction between the two models, that is the YOLO V5 and the DETR, is their respective training approaches. YOLO V5 employs a supervised training strategy that involves anchor-based object localization and classification losses. In contrast, DETR utilizes a fully supervised training approach specifically designed for transformer-based object detection, which includes a combination of focal loss, dice loss, and Hungarian loss. DETR, on the other hand, has shown an average confidence score of 95% on the blood cell dataset. Though the computational time is higher than the YOLO V5, the DETR model performs better than YOLO V5 in detecting small objects in an image as DETR uses a multi-head attention mechanism. Now, the future scope of our project can be improved in the direction of detecting the subtypes of WBCs as well. Again, fine-tuning can aid in adapting the model to the distinct traits and variations of blood cells, improving performance in tasks requiring the detection of blood cell subtypes. Future research may examine methods like model compression, quantization, or distillation to increase the effectiveness of DETR. Finally, we can also work towards improving the interpretability of the model and explore the adaptation of DETR architecture to solve other techniques like segmentation also.